competitive exams, one needs to understand the syllabus properly and organize in a strategic way for the preparation. So I invite my colleague Dr. S. Anju Nair to brief about the syllabus before we begin the discussion.
So the syllabus for paper 2 biochemistry is having 13 components. In the 13 units we have number of subtopics. So before we begin on for discussion to each topic, let me first give an overview of what all topics we are going to discuss. So the unit 1 consists of chemistry of biomolecules. Then unit 2 has DNA replication, transcription, translation. Cell structure and organization is the unit 3. Enzymes, then bioanalytical techniques, then molecular methods, endocrinology and biophysiology, microbiology and molecular biology, genetic engineering. We have a unit on immunology, bioinformatics, and also unit on transcriptomics, genomics, proteomics, and finally the 13th unit, that is cell signaling and cancer. So every unit consists of number of topics, and it is very difficult for us to judge which topic to prepare and how to prepare and what type of questions will be asked in each topic. We will now discuss here today every topic in detail. So first I welcome Dr. Sarada to begin with the unit 1, which is biochemistry of biomolecules.
So looking into this syllabus, it starts with the classification and structure of all the biomolecules. So one needs to be aware of the different types of classification. For example, amino acids, we have structural classification, functional group based classification, essential and non-essential based on their nutrition availability, and similarly for proteins we have general classification based on structure and based on their solubility and other properties, and for polysaccharides also based on their complexity. They are organized as monosaccharide, disaccharide, polysaccharide, oligosaccharide. So for each category one needs to be aware of different examples and also their structures. Similarly for lipids, we have omega classification, we have N based classification. One needs to be aware with the different types of fatty acids, so phospholipids, sphingolipids, sterols, etc. Coming to the next topic, that is biological role of important polypeptides, lipids and polysaccharides, one needs to go with a type study kind. Then moving to the structures of nitrogen bases, familiarly we know the basic purines and pyrimidines, but one needs to know the derived or modified bases, and also one needs to understand their functional role of nucleosides and nucleotides and their basic differences. Moving on to the structure of DNA, apart from the familiar B form of DNA model, one needs to understand the basic difference between the A form of DNA and Z form of DNA, and basics of supercoiling. What is supercoiling, what is linking number, what is rest, what is RIT, what is twist, etc. And also different types of RNAs, like what is mRNA, tRNA, rRNA. Apart from their structure, some unique features of these molecules is very important. Moving on to denaturation and renaturation, we need to know the definitions, we need to know the factors influencing their denaturation as well as renaturation, and some advanced topics like what is C value, what is C value paradox, what is Scott curve, etc. Moving on to the next topic, we have melting temperature or TM value, popularly known as, and then hyperchromic effect, that is absorption of bases or nucleic acids and their susceptibility of nucleic acids in presence of different chemicals like urea, formamide and alkalis, and also enzyme susceptibility. And last topic in this unit is vitamins. So one needs to be aware of group vitamins into fat soluble vitamins, water soluble vitamins, and one needs to understand what is the source of these vitamins, what are the deficiencies or disorders associated with them, and what are the nutritionally rich sources for these vitamins. And this unit ends with the metabolism of all the biomolecules. So metabolic diseases associated with the different pathways. For example, amino acids, we have alkaptonuria, phenylketonuria, maple syrup urine disease, gout, etc. So all these are the basics one needs to understand. So let us just move on to the one example of a So looking into this question, what is the main difference between an aldose and a ketose? So this is a multiple choice question and it focuses on the basics. So one need to be thorough with the fundamentals. So normally students look for either a aldehyde group or a keto group in the choice, but you don't find such words here. Either they are giving a number of carbon atoms, presence of carbonyl group, presence of a hydroxyl group and type of a glycosidic bond. So you logically you have to eliminate, and if you are thorough with the alternative names, so needless to say the right answer will be the presence of carbonyl group. Moving on to the next bit, the alpha carbon of an amino acid is attached to which group? Here the fundamental knowledge of the student is tested. So carboxyl group and amino group are there in the amino acid word itself, but they are also looking for the awareness that the same carbon, 3 different groups are attached. So the answer is carboxyl group, amino group and also the side chain, which actually makes the difference from one amino acid to the another amino acid. Next question can be something like this on supercoiling. So the type of enzymes like topoisomerase, which generally reduces DNA by removing negative supercoiling. So what is negative supercoiling? What is positive supercoiling? One needs to understand. Negative supercoiling is a concept where a tightly coiled DNA molecule will be relaxed by removing some of the twists and turns. So there are different types of enzymes. Topoisomerases are classified based on their mechanism. We have type 1, type 2 and all. So a thorough knowledge about these enzymes will easily make you understand it is type 2 topoisomerase which can introduce negative supercoiling. So we have gyrase. The alternative name of topoisomerase 2 is gyrase, which is popularly cited in all the standard textbooks. Now we have one modal bit from metabolism. This is a very popular disease, phenylketonuria. It is a metabolic disorder caused by the deficiency of an enzyme involved in metabolism of which amino acid? Please concentrate, they are not asking the amino acid enzyme, they are asking the type of amino acid. So phenylalanine, lysine, leucine, tyrosine. Here they are judging this your knowledge on the structure of the amino acid. So phenylalanine is the right answer here. So when you are preparing for the first unit, please concentrate on the structure and the important subtle information about the type of bonds, how they are connected to each other and what are the sources for each disease or disorder. I think now we'll move on to the next unit. I think my colleague Anju Nair will be discussing on the second unit.
The second unit deals with DNA replication, transcription and translation. Now in this we have many subtopics. So firstly, moving on to the process of DNA replication. We all know that DNA is a nucleic acid molecule which is a molecule which is carrying the information for inheritance. So DNA replication is all about how the DNA is going to multiply. Now, how the process in which the DNA replication takes place, it has different steps. There are 3 stages, that is initiation, elongation and termination. We all know these steps and we also know the basic steps, basic components that are involved in each of these steps. But when the questions are framed, when we are preparing for entrance examination, we need to focus on DNA polymerases, we have to focus on the gyrases, we have to focus on the topoisomerases, all these enzymes which are involved in these processes like DNA replication. And also in this syllabus, they are also mentioning about prokaryotic replication and eukaryotic DNA replication. So we need to focus on how what is the difference between a polymerase which is used in a bacteria, what is the difference between the polymerase which is used in the eukaryotes, how many number of polymerases are there like DNA polymerase 1, polymerase 2, polymerase 3, what are the functions of each polymerase. So the questions can be framed on these lines. Also, the next topic, that is genetic code, is another important topic where the questions can be framed based on what are the what are the rules that are followed in the genetic code. That the genetic code is universal, it is having no ambiguity. All these components has to be learnt and understood. Every component of genetic code needs to be understood in order to solve problems based on the genetic code. Similarly, transcription in prokaryotes and eukaryotes and its regulation. So, the DNA to RNA, the process is called as transcription. Now, both the processes take place in both eukaryotes and prokaryotes, but there are subtle differences in the stages like initiation of the transcription process, also post-transcriptional processing. There are a lot of post-transcriptional modifications that take place in case of eukaryotic transcription. And followed by that, operon concept is nothing but a gene regulation concept. Most of the entrance examinations or most of the multiple choice questions focus on operon concept of lac-operon and trip-operon. So, it is we suggest that you should more focus on these two operons. Of course, you should have an overall idea about what operon is, how the regulation of the operon is going to take up. And then we have translation, when the translation is nothing but the process of synthesis of proteins, and the process of synthesis of proteins includes the ribosomes, the mRNA and the amino acids and tRNA. So, we have to have an idea about what type of ribosomes are used in prokaryotic translation, what type of ribosomes are used in eukaryotic translation, and what are the different types of amino acyl tRNAs, what is the initiation complex. Like the initiation complex in the translation process consists of three components, that is, we have the amino acyl F-met tRNA, and then we have a 30S subunit, and then we have the mRNA. So, this is an initiation complex. So, in the multiple choice questions, they may give all the three components, they may give skip one component, give only two components. So, unless and until we are thorough with every step, what is happening and what components are involved, what molecules are involved, it is very difficult for us to eliminate or come to a conclusion about a particular answer. So, also in all these processes, there are some molecules which act as inhibitors, which are called as inhibitors of transcription, translation and replication. It is better to learn some examples about the inhibitors of transcription, translation and the DNA replication. Also, if we look into this question, that is which of the following types of DNA polymerase has 3' to 5' exonuclease activity. Now, this is a sample question as to show how the questions could be framed on enzymes. So, the options given are DNA polymerase 1, polymerase 2, polymerase 3 and polymerase 4. Now, we need to know the functions of every polymerase, structural subunits, what are made up of, what is making up that particular polymerase, and the function of every subunit, so that we will know that one of the subunits has exonuclease activity, one of the polymerase has proofreading activity, and in which direction, 3' to 5' or 5' to 3'. These are very tricky questions. So, you have to have a clarity on which is the direction in which the polymerase activity will take place, or the proofreading activity, exonuclease activity is going to take place. All these needs to be learnt, so that you can easily answer this question. So, you see the answer here is DNA polymerase 1. Now, what is the main enzyme which is taking part in DNA replication is DNA polymerase 3. So, why we have choose DNA polymerase 1? Because exonuclease activity is present only in the DNA polymerase 1, and that to 3' to 5' exonuclease activity. Whereas, in DNA polymerase 3, which is active main enzyme which is having polymerization activity, does not have this. So, unless and until we prepare for every enzyme, its details and its functions, we cannot come to a correct answer. Similarly, we have to look into this question, that is, transcriptional gene control in eukaryotes is mediated by. So, the options here are metabolites that bind to the cis-acting elements, trans-acting factors failing to bind to cis-acting elements, trans-acting factors binding to cis-acting elements, and repressor proteins that bind to operator site. So, the answer here is trans-acting factors binding to cis-acting elements. During transcription, the initiation process what is going to happen will have different steps. That is, you have an initiation where you have a promoter region where the RNA polymerase is going to go and bind to a promoter region, and then the initiation of transcription will take place. Now, during all this, there is a regulation that will happen. So, these regulatory factors are called as trans-acting factors and cis-acting factors. And the regulation can be because of inducers, can be because of the operators which are going to function over there. So, the gene control, the control of that particular gene, whether the mRNA has to be synthesized or not, will be different in different cells. Like for prokaryotic cells, one type of regulation will be there. For eukaryotic cells, one type of regulation will be there. We need to have when we learn, we need to learn the process in both. That is, we have to differentiate and compare and learn. In eukaryotes, this process is taking place like this. In prokaryotes, the process is taking place like this. So, when we learn with such a strategic approach, we will not have any confusion when we are attempting a particular type of question. The next popular questions that come up in biochemistry, in molecular biology, is from genetic code. So, the genetic code is the code which is given for converting the DNA to mRNA, and then then mRNA to finally you have the proteins which are going to come out. So, the genetic code will help us to identify what is the amino acid that will be synthesized as per the codon which is present on the mRNA. Now, you see here the options what we are given here are, there are multiple codons for a single amino acid. There is only one codon for each amino acid. There is a redundancy in genetic code, both A and C. So, as I told, the genetic code has got many rules, around 10 to 11 rules are there. We have to learn what are the features of the genetic code. So, the answer here is both A and C. Multiple codons for single amino acids are there. It is preferable that if possible we can learn some of the popular amino acids and their codons also, because nowadays they are also asking such type of questions also. And the popular amino acids like we have initiating amino acids, formyl methionine, what codon is going to code for that amino acid. Then aromatic amino acids like tryptophan, how many codons are there for each amino acid? Are there 3 codons or are there 4 codons? Lysine can be coded by 4 types of codons. All these can be learnt so that we will have more grip on this particular topic. And then coming to gene regulation. So, gene regulation, as I told, most of the time the gene regulation is focused on lactoperon and tryptophan. So, lactoperon is nothing but the lactose operon, where the cell is going to regulate its metabolism for uptake of lactose and metabolize the lactose. Now, these are here you need to know the concept of inducible genes, constitutive genes. So, if you look here the question, the lactoperon is an example of an inducible operon. Which of the following statements is true about inducible operons? They will when the questions are asked, maybe directly one question may not be asked, one topic may be interrelated with another topic. So, the answers here, if you read, they are always active and continuously express the genes in the operon. They are only active in the presence of a specific inducer molecule. So, we need to know what an inducible operon is, what is an inducer molecule, and why does what is the role of an inducer molecule in an operon. These all concepts need to be thorough. They are always inactive and do not express the genes in the operon. So, what are such kinds of, what is that feature of operon and where does such kinds of operons work? They are constitutively expressed regardless of the presence of an inducer molecule. Now, the answer here is they are only active in the presence of a specific inducer molecule. If we learn at least lactoperon and tryptophan, there are 2 different types of operon which are regulated in 2 different manners. We get a fair idea, because it is not possible for us to learn more than many operons at a stretch. So, at least if we are having these 2 operons thoroughly studied, probably we can answer any of the gene regulation questions. It may not be always direct also. So sometimes the cyclic GMP concepts are also there available in this gene regulation. So also look into these aspects, a little extended regulatory aspects also, how glucose will influence the present, the lack operon. These are all some of the topics that are interrelated with the gene regulation. So look into these topics so that we can have we can have a broader way of thought process working when such questions are asked, especially when they ask which a statement is true. It is very difficult. Sometimes it will be very tricky because most of the statements will be coinciding with the other with small changes. So unless and until we have a very insightful understanding about that topic, it becomes very it will be a confusion for us to choose the correct answer. So therefore, I suggest that when you learn for operon or gene regulation, study thoroughly about these two operons.
Moving to the third unit, we have a syllabus on cell structure and organization. So the important concept in this unit is one needs to understand what is a biomembrane, what is its composition. Like it has both phospholipids and it has to small extent cholesterol in it, only animal cells do have, and then it also has a major component of protein, which further we have a combination of intrinsic and extrinsic proteins which actually define the function of these membranes. So a thorough knowledge of composition of the membrane will help us to understand the fluid mosaic model of functioning. So we need to understand what are different models of biomembranes proposed and what is the advanced model, that is fluid mosaic model, and how intrinsic and extrinsic protein organization is defining the functional role of a biomembrane. So when you go further to electron transport chain and all, you appreciate the structure and function of the biomembrane in detail. Then we have types of transport, which is very important function inside the cell, and we have different types of transports. You will be studying what is active transport, what is passive transport, what is energy associated transport, etc. So one needs to know everything in detail. Then mechanism of two important vital processes, that is muscle contraction, where you will be learning what is actin and myosin role, and also in nerve impulses what is neurotransmitters and how nerve impulses are conducted across the human body. Now we will take some modal questions in this thing just for our understanding. So as a focus, one needs to understand the integral protein or intrinsic proteins, their organization. So you may get a question, what is the domain, structural domain present in membrane? So here the options given are alpha helix, beta sheets, or triple beta sheet, or none of them mentioned. So when we learn the structures of proteins, we will be learning different types of secondary structures and tertiary and quaternary arrangement of these structures. So alpha helix is the ideal structural domain which helps in the transport. So a transmembrane protein which is having more of hydrophobic residues will form something like a barrel kind, and that can be easily constructed with a alpha helix. For example, your G proteins will have 7 heptahelices. So all helical structural domains help in the transport, and also transmembrane proteins or integral proteins commonly have this domain. Now moving on to the next question. Sodium glucose transporter is an example of. So here they are testing our knowledge, like which type of transporter protein is important in this mechanism. So we have on options like what is symport, antiport, facilitated diffusion, and ATP driven. So as I said earlier, all types of mechanisms are given as an option, but we need to know that sodium glucose transporter is a symporter. That is, whenever it is transporting sodium ion, along with it another molecule also get transported. So antiport is in opposite direction, symport is the transport in the same direction. So if you understand the mechanism, then easily you can conclude the answer symport. Now moving on to another modal question in this unit, which one of the following following sets of ions are necessary in the chemical events of muscle contraction? So we need to, if you have a thorough knowledge of the mechanism, so what are the two ions which are important. One is one combination they have given as sodium and potassium ions, which are electrolytes. The other combination is calcium and magnesium ion. Another is sodium and calcium, sodium and magnesium. This is only to confuse us. Electrolytes, sodium and potassium, are nothing to do with the muscle contraction. So obviously we have to go with the calcium and magnesium. Calcium ion is helping for the sliding movement of actin and myosin filaments. If you learn the mechanism in detail, you can easily figure out the answer. Now moving to another modal question in this unit, that is neurotransmitters carry the signal across. So refractory gap, axonal gap, synaptic gap and hippocampal gap. So sometimes people also go with an axonal gap because they remember the neurons have an axon and all. So thorough knowledge on the structure of a neuron will help us to understand what is synapse, and if you understand what is synapse, then it is easy for us to pick up the answer as synaptic. Right? Now we'll move on to the next unit.
So the unit of enzymes, this here it's a very vast unit. Many questions can be framed from this topic, but then strategically we can prepare what are the main areas in which the enzymes the questions are focused are. One area is classification. So classification of enzymes, like EC number of the enzymes, what are the different classes of enzymes, like hydrolases are there, lyases are there, transferases are there, kinases are there, what are the EC numbers for these enzymes, and what kind of work or what kind of function do these particular enzymes perform. So these types of informations if you have, I think a classification and nomenclature of enzymes will be a topic wherein you can attempt any type of questions. Then cofactors and coenzymes is one another important area where the focus has to be laid on the enzymes and their cofactors. Some popular enzymes which you come across in most of the metabolic pathways like glycolysis, TCA and all, we have to learn these enzymes and their cofactors. So automatically when we are preparing for the metabolic pathways, it is obvious that we will learn their cofactors also, but in certain cases it becomes a little confusing when the multiple choice questions are given. So focus on these cofactors and the coenzymes like coenzyme A, acetyl coenzyme A, NADH, FMN, all these types of enzymes, coenzymes and their roles in which type of enzymes. Learn some examples. For every type of coenzyme, learn some examples. So for every type of cofactors, learn some of the examples of enzymes in which enzyme magnesium is utilized. Like glucokinase is there. The first enzyme of glucose to glucose 6-phosphate is there. The different types of important prominent steps wherever you are coming across, learn those enzymes. If not all, we cannot 100% learn all the different types of enzymes and their cofactors, but some popular ones you can learn. Then coming to enzyme kinetics, here some problems may also come up. Like they may give you certain conditions where the enzyme concentrations is a particular concentration of enzyme may be given and a condition may be given, like in the enzyme reaction is suppressed by an inhibitor, then what will be the fate of the product. For this all, mostly enzyme kinetics will focus on, the problems will focus on the Michaelis-Menten constant, that is the Km. So here you can note it down that we should focus on the components like Km value, Km, that is Michaelis-Menten constant, and Vmax, that is what is the velocity or the maximum velocity at which an enzyme is going to function, what is the role of a Vmax, how the Michaelis-Menten constant is influenced by the Vmax, substrate concentration, and Kcat, that is the catalytic number. So these are some of the important clues that I think we should learn and go, so that we can tackle with the problems which are given under the enzyme kinetics, and also learn the different conditions under which the Michaelis-Menten constant will change, that is low substrate concentration, higher concentrations, and the graphical representations of each conditions. When there is a competitive inhibitor, what type of change will happen in the enzymatic reaction. When there is an uncompetitive inhibitor, what kind of change will happen in the enzyme kinetics, and examples of all these inhibitors. Then factors affecting the rate of reaction. So we know that various factors will affect an enzyme reaction, like temperature is there, substrate concentration is there. So under all these conditions we need to learn what will be the change in the enzyme kinetics. Also we have in the syllabus there is a lysozymes, RNAs, chymotrypsin, trypsin and carboxypeptidases. These enzymes will need to be learnt in detail. That is, you have to learn the structural features of these enzymes, active sites, active domains, the domains where the inhibitors will act, and feedback inhibitions of these particular enzymes. And the concept of feedback inhibition is very, very important. And also regulation, that is mainly feedback inhibition is also the regulation. So all the regulatory aspects of all these enzymes which are there in as part of the syllabus. And the type of mechanism, is it a covalent catalysis, is it with an ionic catalysis, all these non-covalent catalysis, acid base catalysis, all these concepts has to be learnt when you are learning about the particular enzyme. So when you are learning, what you can do is you can make a comparative chart, right? You can make a differentiation chart and say lysozymes, RNAs, chymotrypsin, all the three, what is the mode of catalysis. Then what are the enzymes which are, what are the molecules which are going to inhibit these enzymes, what are the conditions at which these are regulated. So all these things can be learnt as part of classification, and you can classify, and as a comparative method you can learn these, so that you will remember the points also, and you will exactly focus on what is really important to understand that particular function of that particular enzyme. Then another important topic under enzymes is laws of thermodynamics. So we know that most of the biological reactions are mediated by enzymes, and here thermodynamics is the change in the energetics of these reactions. So here we have to learn about what is endergonic reaction, what is an exergonic reaction, what is Gibbs free energy, what is entropy. All these context has to be learnt thoroughly, so that we in the examinations, when they give problems based on this, how entropy will change for an reaction which is reversible, what is the free energy change for an irreversible reaction. All these kinds of questions may come up. So in the next slide I will also discuss some of the questions based on this, so that you will get an idea. So whatever we are discussing here, they are only a hints for you. You have to note down these areas and these top points and elaborately learn on these particular topics. Then coming to biological oxidation. So here biological oxidation, high energy compounds, these are some of the topics where questions may be asked as like what name among the 4 given molecules, which are the high energy compounds. And for biological oxidation they may ask NADH molecule, how many electrons will it transfer for a particular reaction, or enzymes which are involved for the biological for that particular coenzyme or cofactor, coenzyme where the biological oxidation is taking place. All these things can be learnt in the form of some examples. So if you learn one example, similarly you can interpret the answer for the other answers or other questions which are being given in the examination paper. Also electron transport chain is one important area where you will have to learn about redox potential. And electron transport chain in both eukaryotes and prokaryotes, there are changes, there are variations in eukaryotic complexes, electron chain complexes, also prokaryotic electron chain complexes. So we know that electron transport chain is lined up with the complex 1, complex 2, complex 3 and complex 4, which is ATP synthase. Now in each one there is a mechanism, there is a redox reaction which is going to take place. A component is going to get oxidized, a molecule will get reduced, how many protons are liberated for every reaction, all these needs to be learnt. Also we need to learn about number of ATP molecules which are generated at every step of electron transport chain. And what are the inhibitors which are going to inhibit the electron transport chain? There are various inhibitors which will inhibit the like amphotericin, etc. They will inhibit certain molecules or certain complexes inside the electron transport chain. And also we should know how do we connect the electron transport chain with various other metabolic reactions. From TCA cycle, which component will travel to the electron transport chain? From the glycolysis, which molecule is going to enter into the electron transport chain? What is the mode of the functioning of electron transport chain? All these regulatory aspects also, if you go in detail, I think this topic can be easily answered. And then coming to photosynthesis, we have to learn both in higher plants, also we have to learn in photosynthetic bacteria or algae. So we have Calvin cycle, we have cyclic phosphorylation, non-cyclic phosphorylation, learn about the transfer of electrons. Say for example, they may ask, the pheophytin takes electron from which electron carrier? So we need to learn the sequence of the electron transport chain, or in the photosynthesis also, whether it is cyclic or non-cyclic photophosphorylation. We have to learn in different organisms like blue-green algae, cyanobacteria, that is blue-green algae, and also we have to learn in higher plants. So oxygenic photosynthesis, non-oxygenic photosynthesis, what is the difference between each of these? All these need to be focused on when we are learning about photosynthesis. Also the pigments, photosynthetic pigments which are going to be used by various organisms, where that is prokaryotes as well as in eukaryotic plants also. There are different types of photosynthetic pigments, their structures, right, the porphyrin nature of the chlorophyll and the functioning of the chlorophyll. All these things are some of the wide areas where people can be focused with certain questions. Now we will move on to some of the questions based on this unit, that is enzymes. So here we have which of the following coenzyme does not function as a redox reaction? So I already told you, you need to know what a redox reaction is. So what is a redox reaction here? When a compound is accepting electrons, it gets reduced, and then when it is releasing the electrons, it is oxidation. So in these molecules, which of the following can happen? Which of the molecules can undergo or can participate in one such reaction? Here flavin adenine nucleotide. When we have learnt many biochemical pathways in an electron transport chain, etc., we see that FAD is getting converted to FADH2. So we know that yes, FAD and oxidized form has become an FADH2. That means flavin adenine nucleotide cannot be the answer. Then go for deoxyadenosyl cobalamin. Probably many of us, we don't know also what this molecule is. It is not very frequently encountered in many of the reactions. Then coming to nicotinamide adenine dinucleotide. So one very important thing that I would like to tell from this type of question is, don't just learn the short forms like FAD, FMN and all, but also learn the expanded names of these particular molecules, so that if in whichever form they are given, instead of flavin adenine dinucleotide, they are giving it as FAD or FMN, whatever it is, you should be able to figure it out. So nicotinamide adenine dinucleotide NAD gets reduced to NADH2 in many reactions, like for in TCA, etc. So NADH2 is possible. Flavin mononucleotide FMN also will be converted. So the only by elimination we can say that deoxyadenosyl cobalamin could be the answer. Though you may not know about the function of that, or you don't know where exactly this molecule might be working in a metabolic pathway, but then still you can figure out the answer by knowing the other answers, by reasoning the other options given, you can probably come to a conclusion in the answer. Then coming to the functioning of enzymes, that is enzyme kinetics. Here this is one of the type of questions that people may feel little confused when they are reading it, but actually this is a very easy question. Look here, when the velocity of enzyme activity is plotted against substrate concentration, which of the following is obtained? Now whenever we are reading a question, we should first find out what conditions are proposed in that problem. Here they are asking when the velocity of enzyme activity is plotted against a substrate. That means you need to put a graph on the paper and check. It is a plot, right? So you have to plot and see one side velocity and the other side so on y axis you put velocity, on the x axis you put substrate, and see if which of the following is obtained. So if the velocity of the enzyme is increasing, naturally the velocity of enzyme will increase as the substrate concentration will increase, but only up to a condition where the saturation of that enzyme will take place. Once the active sites of an enzyme are occupied by the substrates, how much ever substrate you will add into a reaction, it may not matter much for the change in the product formation, that is rate of product formation will not get altered. So in such a condition, when you are putting a graph and when you are plotting this condition, you will know that it will come up as hyperbolic curve. It cannot be a parabola, it will be a hyperbolic curve. Like this, if you know the different conditions under which enzyme activity, how it will happen, you can easily configure the answer whichever type of problem is given. And then we have what is the value of delta G when a system is in equilibrium. So here you see the options given, delta G is equal to 0, delta G is Gibbs free energy. I told you Gibbs free energy, entropy, enthalpy, all these concepts need to be learnt so that you can answer such questions on from thermodynamics. So delta G is -1, delta G is equal to delta G. Now what does it mean by an equilibrium system? So you imagine a reaction which is in equilibrium. Reaction has two components, a reactant and a product. So if both are in balance, that means the reaction must be an equilibrium reaction. So rate of formation of product is equal to rate of formation of a reactant, means it is a reversible process. So when a reversible process is in equilibrium, it is in equilibrium. In a reversible process, delta G will be equal to 0 because there is no free energy exchange. No, one side of the reaction is not getting the free energy because the rate of formation of the product and the reactant both are same. So therefore by reasoning we can come to a conclusion that this is delta G is equal to 0. Then coming to a process by which the pH gradient across the membrane provides energy for synthesis of ATP is called as. So most of the times here we have to learn certain terminologies. What is phosphorylation, what is transfusion, diffusion, chemiosmosis. Now when I am talking about an energetic mechanism across a membrane, naturally it cannot be diffusion because it is not an energy dependent process, whereas in case of transfusion it does not happen. Now so we can eliminate these two options. Coming to phosphorylation, phosphorylation is a process that is going to take place by when ADP is added to a phosphorus group, ATP is formed, which is phosphorylation. Now coming to the fourth option, that is chemiosmosis. Here the pH, now in phosphorylation there is nothing relative to that which is relative to pH gradient, but here if you see chemiosmosis, it is a process which is which is developed because of both the chemical gradient as well as the ATP formation is also seen, that is the energy formation is seen. So here the chemiosmotic theory, which is part of the electron transport chain. This is a question from that particular topic, and the answer is chemiosmosis, which can be easily understood from the words which are given there, that is pH gradient across the membrane, the providing formation of energy, which happens only in case of chemiosmosis model. Also we should learn some of the main important theories proposed by whom, the certain scientists, like chemiosmotic theory proposed by Peter Mitchell. These questions can also be asked because they are very popular and Nobel Prize winning discoveries. Then coming to which of the following factors does not influence electrophoretic mobility. Now here you can see that the stereochemistry of molecule is going to come into picture. I think this is a topic which is part of the bioanalytical techniques. Here bioanalytical techniques is one of the unit in which you have to learn about various techniques like chromatography, centrifugation, etc., which Sarada madam will discuss. So madam, over to you.
I think the fifth unit is bioanalytical techniques. So this unit has all the techniques like calorimetry, spectrophotometry, and then XRD, and then you have NMR, you have MS, AAS, that is atomic absorption spectroscopy, flow cytometry, and all the techniques which normally you have centrifugation, you have electrophoresis, you have chromatography techniques, all the techniques which are placed in this unit. One needs to understand three things. Any technique, any biochemical technique, three components are important. What is the principle based on which the technique is operated? What are the components, a design of the instrument, and what is the flow, workflow, or what is the flow chart of it, and then what are the applications, when and where this technique is used. And some techniques you have to also know the advantages, limitations, and disadvantages. All these are important. So I advise all the aspirants to make a chart. Write the technique, write the principle, write the important design, which are the components required, and then move on to the applications. If you can make a small what you call concept map for each technique, that will be helping you for the preparation. So here we'll take up some questions like this. This is something on electrophoretic mobility, which is very commonly asked. It's since I said it is a model, so they will confuse you and they want to understand your understanding of principle. So factors which influence, which does not influence, is the key thing you have to remember. So molecular weight or size or shape do influence the mobility of a molecule in electrical field. So that which is not influencing is only the stereochemistry. So and if you go with a logical analysis, you can understand that it is stereochemistry. Now another question from this unit could be something like this: which of the following techniques would be most useful in identifying as well as quantifying the presence of known impurity in a drug thing. So here again, if you are familiar with each technique and its application, then you will easily rule out. So NMR is for structure prediction. Mass spectroscopy is also understanding for structure prediction, and sometimes you can also go for molecular weight determination. Infrared is nothing to do with the purity thing, but HPLC will give you such a analysis where you will you can know the concentration of the component, and also the peak analysis will give us the presence of impurities. So presence of a known impurity in a drug substance can be easily figured it out with the technique called HPL, so high pressure liquid chromatography. And nowadays we are getting lots of advanced techniques also, like FPLC, HPTLC, and all that. So I advise everyone to understand the variation of each technique and what is it used for, what is its application, and also the limitations, because some techniques cannot be used for all the purposes. So some techniques are used only for like your IR and MS, right? So moving on to next unit, molecular methods. Over to Anju.
Molecular methods is a unit wherein we have topics like PCR, real-time PCR, fluorescent in situ hybridization technique, RFLP, RFAD, DNA fingerprinting, etc. So when we talk about these techniques, most of these techniques are application oriented. When we learn, we need to learn about the principle of these techniques. We need to learn about the mechanism in which this technique is done, the flow chart. What we can do is, say for example, PCR is there. You have to understand what exactly PCR stands for. The expanded forms need to be learnt, and the short forms of the techniques also should be learnt. And then we have to proceed on to how this particular technique is going to benefit. What is the purpose of doing this technique? Like PCR is for amplification of DNA. Now what are the areas where this PCR technique is going to be used? Like you have for R&D purpose, where you want to amplify a piece of DNA. It could be a recombinant DNA, or in diagnostics where you need to amplify the DNA samples, or in case of forensic sciences they may be used. So application of every technique is very essential. As far as the procedure is concerned, what you can do is write down a flow chart, which will be the easiest method to learn a technique. So flow charts will give you a very crisp idea about how the technique is going to take place. Within PCR, again we have different types of PCR. Real time PCR. So in advanced since it is an advanced technology, questions may appear based on real time PCR. So here we need to differentiate between both of these PCRs. In real time, we know that it is a fluorescent technique, wherein before the technique is completed, that is as the technique is being proceeded, as the reaction is going on, we can take the real time picture, or we can take the real time data of that particular reaction at any point of time when the reaction is going on. And the use of this kind of real time PCR, in what areas are they going to be used. Similarly, in situ hybridization, fluorescent in situ hybridizations like fish, RFLP, RAPD, the full forms of RFLP, restriction, fragment length, polymorphism. What are these? What exactly these mean to us? Where are these present in the DNA sequences? What do they signify in the DNA sequences? In what way we can exploit these sequences to understand various diseases, to screen out what are the different types of malformities, deformities that are happening, what are these repetitive DNA sequences, what are present. All these can be understood by doing these techniques like RFLP, RAPD. And DNA fingerprinting, I think it's a very popular technique, so therefore you can also expect some questions from them, but not directly. They may ask you certain conditions, certain steps in a particular reaction, and they may ask you what technique it is. Is it a DNA fingerprinting technique? And DNA microarrays, a very advanced technology in case of diagnosis also. So this is nothing but biochip methods. We need to learn about the ligands and various diseases which are diagnosed by this DNA microarrays. What is the role of a ligand on a biochip? What is the role of, what type of DNA microarrays are present? Examples of, some of the examples you can learn. Then coming to DNA sequencing. DNA sequencing is a topic where you have traditional methods, which is an older method, and DNA sequencing we have advanced technologies which are bioinformatics based. So we in the traditional experimental methods like Edmonds reaction, Sanger sequencing method, Gilbert sequencing method, all these methods are there, Maxim Gilbert sequencing. We have to learn, put a flow chart and learn how these techniques are done, what are the reagents used in them, like a paradinitrophenol or whatever the type of reactions in Edmonds reaction, what is the type of reaction, C-terminal sequencing, N-terminal sequencing, which one is what, all that you have to learn. And coming to DNA sequencing in advanced methodologies like new generation sequencing, right, bioinformatics based methods of DNA sequencing, all these have to be updated. So don't just go by the older versions, also focus on the newer versions. So next generation sequencing, these are all the new topics. So focus on the new topics, so that the questions that may appear could be because of the current usage of these particular techniques may have these questions. So not only that, you have DNA probes. Now DNA probes are used for various techniques. So DNA probes are used in DNA microarrays, etc. You should learn what is a DNA probe, what is the length of the DNA probe that is used, what are the features, and sometimes there are also DNA probes that are designed in silico designing of the DNA probes. These are all some of the topics, advanced topics that you may read and you may go. Also we have short videos, animations which show only that particular topic. Like for example, if you click on DNA sequencing by Maxim Gilbert, small short animated videos will be there where only that particular method will be shown. So try to focus on such kind of videos and prepare for these topics. Also mainly learn applications for all these topics. Coming to blotting techniques include western blotting, northern blotting, southern blotting. What is northern blotting, what is western blotting, what is immunoblotting technique. all these can be learnt with reference to application as well as what is the purpose of each one and how it is done. So, if you are done, if you learn these many aspects, I think you can cover some of the questions related to these topics. So, let me cover some of the questions also in these areas. What is the purpose of PCR product in Sanger sequencing? In the options here is given to amplify the DNA, to sequence the DNA, to cut the DNA, to isolate the DNA. So, you need to know what exactly is Sanger sequencing and what is a PCR product. And I also missed, when you are learning PCR and other techniques, you should learn about various conditions also. What is the temperature at which the technique is happening, what is the annealing temperature, what is the denaturation temperature. So, when you go such deeper aspects, you can answer such questions. So, the answer is to sequence the DNA. So, unless and until you learn that topic with specific goal of learning the principle, learning the application and also learning the mechanism, you cannot remember exactly which technique is used for what and what happens in every technique. So, this is how you can configure the answer. Now, when we look at such questions, here you see which of the following is incorrect about oligonucleotide design in a microarray. So, first of all, you should know how a oligonucleotide is, what is an oligonucleotide, where and for what purpose the oligonucleotides are made, and in a microarray why an oligonucleotide is used. So, if you know these concepts, then only when we can have a clear picture of which option is correct or incorrect. So, here the answer you see, the oligonucleotides do not react with cDNA samples, which is wrong. The oligonucleotides do react with the cDNA molecule, which is a complementary DNA molecule. Unless and until the oligonucleotide attaches with the cDNA molecule, it can, that ligand binding, if it is not taking place, further hybridization of the sample or test sample will not take place. So, to understand this, you should understand exactly how DNA microarrays work. So, such questions need to be focused on and learn the topic as per the questions that we are showing. So, which of the following is a benefit of next generation sequencing? So, I told you any question which is related to new techniques will be mostly focused on in the current entrance examinations, and next generation sequencing is the today's hot topic. So, therefore, all the above is possible with next generation sequencing. So, kindly update with all these topics also. Now, when we move on to the next topic, Dr. J. Sarada will brief on the next topic.
Yeah. Moving on to this part of the syllabus, endocrinology and physiology. I think it begins with the organization of endocrine glands, their location and their function, then classification of hormones, like what are peptide hormones, what are steroid hormones, then structure and organization of muscle and nervous system. Again, in earlier units also we had this mechanism of muscle and muscle contraction and nervous system. So, these are little interconnected. A thorough knowledge in these areas will fetch you good marks there, and abnormal electrolyte composition of blood, that is important, and again major analysis of cardiac, liver and thyroid diseases. And you will be you have to focus on the structure, the quantity of hormone, type, nature of hormone, and what is the conditions which will influence this hormone synthesis, etc. So, disorders of kidney and things. So, we will take some modal questions in this category. First question concentrates on which of the following symptoms would be expected to cause the goiter, caused by goiter. So, goiter is a here you will be given a condition like what is goiter and what is the factors which normally people understand how the T3 and T4 hormones are regulating here. So if you look into all the 4 options, so you will they will confuse you by giving the different conditions, whether is there any weight gain or weight loss, is it any symptoms expressed or not, like that. So if you are really reading the content thoroughly, then it is very easy for you to understand the condition of hyperthyroidism, elevated levels, which will cause in weight gain, not the weight loss, and also a symptom of cold sensitivity will be associated with this. So a logical reasoning is important. And coming to alcoholic liver disease, here they are asking for the marker enzyme, which enzyme is sensitive marker for this condition. So if you read, if you are little careful, alanine transaminase, aspartate transaminase, and all transaminases normally they work on the transfer of amino group. It is the aspartate transaminase which is whose metabolism actually happens inside the liver. So that is the right answer. And this, the sodium in the kidney is reabsorbed. So in this bit they are checking your knowledge on the structure of nephron, and what are the which part actually the reabsorption of the electrolyte is happening. So the answer as renal tubules. Next bit. So we'll move on quickly from now onwards with the units. So I'll ask Anju to continue.
So the next unit is microbiology and molecular biology, where we some of the topics deal with the bacteria, viruses, the replication of the viruses, some of the animal viruses, plant viruses, popular animal viruses and plant viruses, and gene organization and regulation in prokaryotes and eukaryotes. So moving on to the type of questions that can come over from this particular unit, here you see what is a transduction process. So this is a part of gene transfer mechanism which is seen in case of bacteria. You see here the genome is transferred from one bacteria to another bacteria. The part of the DNA is transferred by bacteriophage from one bacterium to another bacterium. Is it a plasmid which is transferred from one bacterium to another bacterium? So there are 3 different types of gene transfer mechanisms, like transduction, conjugation and transformation. These are the 3 areas where the questions can come up. So if you know thoroughly that transduction is a process mediated by a bacteriophage, which is a virus of bacteria, and transformation is uptake of naked DNA, and conjugation is the transfer of gene from one bacterial cell to another bacterial cell by means of a conjugation tube, I think these type of answers can be easily noted. I mean, you can easily get through such questions. So look into the topics of gene transfer mechanism, and as the answer shows here, the part of DNA which is transferred by bacteriophage from one bacterium to another. Sometimes the questions will be a little bit elaborate, so, but if you know the topic, it will be very easy to identify the answer. So now this is a topic from adenovirus. So there are some viruses lined in the syllabus. Go through that particular virus structure, their replication mode and what they cause. So you see here what are the correct statements about adenovirus. So you need to know whether the virus is a DNA or an RNA virus. So do they contain an envelope or not? Is it an enveloped virus or a not enveloped virus? What type of infection it causes? Here they are giving it causes respiratory infections, resistant to environmental factors. So the answer here you can see it is the third option. That means second, third and fourth options are correct. It is not a RNA virus, adenovirus is a DNA virus. So in such way, if you learn, focus on the specific subjects of every virus, you can easily answer these questions. Then coming to nucleosome is made up of. Now this is a question based on the genome organization, wherein you have what a nucleosome exactly is going to be made up of in a chromosome. Here it has given 4 options, out of which the answer is DNA, histone core protein and linker H1. So there are different histone proteins also, significance of different histone proteins. So you have to learn on what are the different types of histones, how they are organized, what are linker DNAs, how the chromosome is wound, I mean how the DNA is wound into a chromosome and packed in a cell. All these things has to be learned in genome organization. And then we move on to genetic engineering, which madam will deal.
In this unit, it starts with the enzymes, that is your invisible molecular scissors restriction, which will actually cut the DNA. So, one needs to understand the classification as what is type 1, type 2, type 3, and also the advanced artificial restriction endonucleases also, like you have your talons and Zephyrs and other things. So, cloning and expression vectors, we have different designs right from the cosmate, backmate, artificial chromosomes, and also shuttle vectors and advanced techniques. And cloning methods, we have these methods are different. For prokaryotes, we have a different strategy. For eukaryotes, again for yeast, it differs from one organism to another organism. So, we will take a model question here. The restriction endonuclease is having a defense mechanism in the bacterial system against foreign DNA such as viruses. So, but how it is able to protect its own DNA. So, they are asking the basic thing, like how virus is protecting itself and it is cutting only the bacterial DNA. It is because it is understanding the modification which is happening in the bacterial DNA, that is by methylation. So, methylation of bacterial DNA can be recognized, and that is how the restriction enzyme will recognize and cut it. So, you have to understand which type of modification is important for the recognition here. Yeah, next bit. So, which of the following properties is not taken into account while looking into the suitable vector? So, when people are selecting a vector, now it's not simply pick up any vector and start for cloning, but you have to understand the carrying capacity of the vector, that is the size, the organism in which the vector is being put in, that is the host, and also the restriction site where actually it is, and is there any autonomous replication property, that is origin of replication. So, here out of all the 4 options given, it is the which is not taken into account. So, again one has to read the question properly. It is, we are not bothered in which parent organism the vector is being expressed, but one has to bother about the carrying capacity, size, restriction and ORI sites for the vector design. So, which of the following statements is false? So, one has to keep in mind that there are different types of logical, analytic and based questions. So, the cells which take up the recombinant plasmid, I think 4 options are on the screen. So, which of the following statement is false? If you really go through the content thoroughly, you will easily come to the conclusion that cell which take up the recombinant plasmids are drug resistant, because the plasmid will have this marker. So, the cell which is gaining this plasmid inside or taking up, if it automatically will have the property of drug resistance. So, next unit.
Then a unit which is going to be discussed the next is immunology. Now, the immunology has some basic concepts in immunology, like what are the components of immune system. As you all know, immune system is the one which is going to protect from most of the infectious pathogens, and we have to learn about theories of theories of antibody formations. We have to learn about the humoral and cell mediated type of immune response, what is a complement, hypersensitivity, autoimmunity. These are the major areas which will be focused in the exam. Coming to the questions, by discussing the questions, let me throw a light on what type of areas and what type of questions will be asked. Here you see which of the following is a primary lymphoid organ involved in development and maturation of T cells. So, to answer such questions, you should know what exactly is a lymphoid organ, what are the components of the immune system. So, here you see spleen, thymus, lymph nodes and appendix. So, there are primary lymphoid organs, secondary lymphoid organs, and these organs are useful for certain function in the immune system. So, we need to know that the thymus is a primary lymphoid organ. It is the site where the T cells will mature, then we can answer such questions. Similarly, all the types of components which are there, you should learn their structure, function and exactly what is the role of those structures and those cells in the immune system. Then such questions can be answered. Now, if you see here, the which antibody isotype is primarily involved in allergic reactions. The options given are IgA, IgD, IgE and IgG. These are the types of immunoglobulins. So, types of immunoglobulins, their structure, their functions and their applications. When you understand, you can easily know that IgE comes for or is raised in the body fluids for allergic reactions. Coming to the next question, which of the following is an example of a systemic autoimmune disease? So, autoimmune diseases, you have to learn all the autoimmune diseases, and most popular autoimmune diseases, localized diseases as well as systemic diseases needs to be learned. So, systemic lupus erythematosus, it is one of the disease which affects the entire body. So, such questions will focus on what type of the disease it is. It may also ask which among them is an autoimmune disease. So, you have to learn all the autoimmune diseases and their effects or their manifestations. Yeah. Moving on to the next topic, bioinformatics.
We have 4 topics in this unit, that is biological databases. Here one has to get acquainted with what are primary databases and what are secondary databases, and also a knowledge thoroughly on what are different types of DNA databases and protein databases. Then you will be learning the technique of pairwise alignment, then MSA, that is multiple sequence alignment. Again you have different techniques for this, and BLAST and other things. Then you have SNPs, that is single nucleotide polymorphisms. So just as a model question, we'll take this: which database provides information about the metabolic pathways and associated genes. So here they're checking our knowledge on different types of databases. If you are familiar with the primary and secondary databases, you can easily rule out that GenBank cannot be. UniProt and String belongs to the protein databases. So you can easily zero it down to the KEGG, which is actually a database which connects different pathways with molecular interactions and other things. So here I advise everyone to make a chart and mnemonic so that you can easily organize all the databases and make a flow chart and understand how you can do the analysis. The second question from this unit could be something like this: which of the following is the primary goal of multiple sequence alignment? So they will simply confuse you by giving different types of options, either to identify the conserved regions, or is it comparing for similarity, or you are going for the sequence to identify the similarity and differences, or you are simply predicting a structure out of it. So here the aim is which of the primary goal for multiple sequence alignment. First of all, we are looking for the similarities and also the differences. So the answer could be something this. Now another popular question on SNPs could be how can SNP be used in personalized medicine? I think single nucleotide polymorphism is a very popular aspect in bioinformatics as well as in molecular markers also, because they they are used for different types of diagnostic purposes. So these are required to develop targeted therapies for specific genetic diseases, that is, and also personal understand the evolution. So SNPs can be used to develop a targeted therapy or personalized medicine for a particular genetic diseases. Moving on to the next unit.
Transcriptomics, genomics and proteomics, these are topics which are of current importance, and transcriptomics, genomics, proteomics are an extensions of the transcripts, that is the mRNA functions, and the omics part is nothing but the computational aspect of it. So if you see here, the most important topics covered under this is gene silencing, protein arrays, and most important techniques like MALDI analysis, which is done for protein analysis techniques. We will directly move on to certain questions which could be asked on them. So the antisense oligonucleotides, that is also shortly called as ASOs, that target mRNA molecules are typically. So these are the technologies that are used for silencing, gene silencing, and for targeting certain diseases also, cure of certain diseases. Personalized treatment of diseases is also targeted using such antisense oligonucleotide techniques. Gene therapy, which is called as, so we should know that single stranded DNA molecules are going to act as antisense oligonucleotide, and we need to also understand the mechanism of how these are going to silence the mRNA. Then coming to which of the following protein motifs is involved in DNA binding. So the DNA structure and the DNA motifs. Motifs are nothing but the different configurations and the structures that are going to happen when the DNA is in different conditions. So zinc finger is one of the answer for this. So we need to learn about these different motifs, protein motifs which are involved in DNA binding. Which of the following is typically used as energy source in MALDI analysis? Here MALDI analysis is a technique which is used for deciphering the protein, identifying a specific type of protein. It's a very rapid analysis and it is a matrix associated laser dissociation technique, and this technique involves UV laser. So if you know the expanded form, I hope it will be very easy to decipher the answer. So sometimes we ignore what are the full forms of the tests or techniques. I suggest that sometimes it will be very useful for us to easily understand which is the answer. And moving on to the last unit.
Let me just quickly wrap up on this unit. We have cell cycle and regulation. So one has to understand the phases of cell cycle like G1, G2, S and M, and then what are the cyclin dependent CDKs and different types of cyclins which will regulate this cell cycle, and checkpoints also we have. And moving to tumor promoters and tumor suppressors. So you have oncogenes and tumor suppressor genes like your RB gene, P53 gene, MYC gene, etc. And stem cells, cell differentiation, fibroblast and muscle differentiation. So one needs to have a thorough understanding what are different types of stem cells and how to propagate them and how to maintain them in the laboratory, and what are their applications, or what is a stem cell therapy, some basic idea about the therapy models, methods also one needs to keep in mind, and all the tissue culture methods one has to understand the types of media, types of propagation and temperature conditions where they are cultured. And the last one focuses on cell signaling, and here we have different types of classification like secondary signaling molecules or second messengers like your cyclic AMP, cyclic GMP, nitric oxide and DAG, IP3, etc. Then G proteins are which are very important in signal transduction. You have to understand their structure and functioning and some cascades. I think you will be understanding different types of signal transduction mechanisms. So we'll take some model questions here. Here in this is a big unit. So you have a mechanism of programmed cell death or apoptosis, where one has to focus on intrinsic pathway and extrinsic pathway and disorders associated with it. And coming to cancer, you have to understand proto-oncogenes and oncogenes. Here I think there are many literature or textbooks where you have tables and also charts available for you to understand type of cancer, which gene is responsible, what type of marker they use for diagnosis, and what are the conditions associated. A good concept maps will definitely help you for the cancer part. Cell stress signaling in plants and plant hormones, and also the last one is your animal cell culturing and their applications. So some questions in this cases, they'll be asking you the protein complex which is used for removal of cyclins. So cyclins are proteins, molecules which will associate with CDK and regulate the cell cycle, and they are not permanent inside the cell. They come and they vanish. So there is a complex called APC complex which actually regulate their concentration. So if you really are thorough with the mechanism, so it is easy to figure out that NFS promoting complex APC will eliminate these cyclins inside the cell. Now second question will be, which type of receptor is involved in signal transduction through the cell membrane. So because all your peptide hormones, they have receptors on the membrane. So GPC related receptors only can catch these signals or messages that is from the hormones, and they can interact and they can send the message. So it is the right answer will be GPC here. Not an intracellular receptors are for steroid hormones, and enzyme link receptors or ligand gated ion channels cannot take these messages. So the answer is very clear, it is GPCRs. Next, which plant hormone regulates the stomatal closure? I think if you are thorough with the different types of hormones, here also a chart on hormone, types of hormones, their structure, function and will easily help us to understand it is abscisic acid, which will regulate water stress. What is the purpose of trypsin in cell culture? I think this is a bit testing our knowledge on cell culturing methods. So, the role of trypsin is to detach the cells from the culture surface. So, when you are culturing on the monolayer, then a unique petri dishes of cell culturing, when you learn, you will be understanding the role of each important ingredient. So, I think very quickly we have discussed on the all units of paper 2 biochemistry syllabus. I think this discussion could be of some help to all of you for your preparation. Wish you all good luck, and if you need to know something in detail, I think we are here and we can take up for further discussion also. I think we wish all the best for all the aspirants for this exam. Thank you.
Wishing you all the best for your preparation of the entrance examination. Thank you.